Hello everyone, today we're at it again and we're doing another BGCSE review. Today is biology and we're doing a paper 3 and this is from the year 2018. Now remember, always write your school number, your candidate number, very important. Also write your surname and your initials, very important. Again, always read through the instructions carefully and also follow the directives of the examiner. Now let's go into our first question. And question 1 reads that the question involves an investigation carried out by students on collecting data on an ecology field trip to a rocky shore. The diagram shows the apparatus that they used. And so let us observe the apparatus at this time. And so the apparatus is this. It contains some subdivided strings. It also have a wooden frame or a metal frame. And of course, you have some species inside. All right, so let's go to what the question is asking us. And so the first part of the question reads, it says, what is the name of this piece of apparatus? And this is a 1-mark question. And this is called a quadrat. This is called a quadrat. All right. So now let's go to our part 2 of this question. It said, what is this piece of apparatus used for? All right, so let's talk about what it is used for here. All right, so starting that. All right, so it is used to estimate the number of stationary or slow moving organisms in an area or ecosystem. Okay, in an area or ecosystem or habitat, whichever one you want to put. Okay, so that also works. In an area / habitat or ecosystem. Okay, let's put it right there. All right, so the bottom line is you can only use it to measure the organisms that are stationary, generally plants and so on, and also organisms that are slow moving. All right, so let's go to part 3 of this question here. Now, let's briefly describe how this piece of apparatus is used. So how do we use this apparatus? So the 2-mark question, again, look at the marks and use them to guide or gauge your answer. And so the first thing I would mention right here, let's put it there. The first thing is that it is placed randomly in a given area or ecosystem. So it is placed randomly. It's placed randomly in the given area or ecosystem. And of course, it's best to do it randomly because you get to have a better count, or at least a more reliable type of survey. Okay, so it's used as a surveying to count number of species or organism in that population. All right, so again, the next thing you want to do is the number of organisms is counted. So the next step you do after you toss it randomly, place it randomly, is to count the number of organisms. Okay, so the number of organism or the specific species that you're dealing with, you just count them. Okay, so the number of organisms are counted. All right, and you can, a matter of fact, just to make a note of that, you only count the organisms that are present within the squares. Okay, so if you go back to the diagram, you'll see the squares. I must go back real quick. So you only count the organisms that are found within the quadrat itself. Okay, All right, so just an estimate of or a sample size, estimate of a sample size, and you can use that to predict a larger area. So part 4 now is saying that this apparatus works better with periwinkle rather than crabs, and explain why for 1 mark, and simply because the crabs may move out of the square. Okay, so the bottom line here is that the crabs may move, and generally they actually will move, because once they toss something over them, they want to run away. Okay, so they will move out of the square. Okay, so very important. Out of the quadrat, you may want to say out of the quadrat or square. All right. All right, let me just put our quadrat. All right, so that's one first point that you get right there. So the move out of the quadrat, move out of the square, right? And just to make a note that the periwinkle will remain stationary, because again, they do not move fast and they tend to be in one place. Okay, so the periwinkle, just to put that in, the periwinkle will not move, or they will stay stationary. Okay, will not move or remain stationary. All right, let's real quickly. All right, so that's exactly what would happen. That's why it's best for use for periwinkle rather than using for crabs, because the crabs they move really fast, or relatively fast. So definitely, if you toss the quadrat, they will run away. All right. Notice when you go to the rocket shore and see some crabs, as they start move towards them, they run away. So imagine you toss the quadrat, they will be moving so rapidly. All right, for this question now, I have this gray background to make it easier to see these organisms. All right, so bear with me with this. All right, so this one now is part B of the same question. It said the diagram below shows a survey which can be done to investigate organisms found living on a rocky shore ecosystem. So we have 3 samples: sample A, sample B, sample C. Now here it asks us now, well, not asks, but it's showing us that we have barnacles there, we have the apparatus, and we have starfish. So there are 2 highlighted organisms so far. We have other organisms. If you notice, you see a sea anemone right here, sea anemone, that is present right here. And we have some other things such as algae and all that, right? So we'll see exactly what they ask us. Now, the formula given here said density is equal to total number of individuals counted in samples A, B, and C over number of samples multiplied by the apparatus area, or the area of the apparatus. Now, if you notice here, right, that the total number of individuals counted over number of samples, that actually give you average, right? So the next formula for this could be the average divided by the area. Okay, so I could actually summarize this formula. So I'm going to say this could be the average, okay, over the area. So let's put it right here, okay, over the area. So that's actually another formula to do this. Then put a line through it. All right, so this could be another formula. So again, total number over number of samples, give it the average per sample, and then you divide it by the area, which is the area of the apparatus. Okay, so just that is a summarized formula. All right, so now let's go down to the question. Now it said, if the area of the apparatus is 0.25 meters square, use the formula above to calculate barnacle density. And when we talk about density, talking about how dense the population of barnacle is. All right, how many organisms are there, or how many number of that species are present in that particular area. Okay, and so let's jump into the barnacle. First, you have to count the total. Okay, you need the total number. We have the number of samples, which is 3. Okay, so let's see if we could get the number of barnacles. So let's count them real quick. All right, so barnacles, if you count this area, this is 3 right here and 3 right here, that's 6. Okay, so we have 6 barnacles there. All right, so let's quickly mention that. We have 6 barnacles here. So barnacles in this area would be 6. Let's put 6 up here. Okay, so the number of barnacles in this will be 6. For this one, number of barnacles will be 3 right here, 3 right here, and 1 there. So there'll be 7. So estimated of 7 barnacles in this one. I know it may be a little bit difficult to see, okay, but I'll zoom in for you a little bit afterwards. All right, for this one here, in all of this region, if you count them all the way, 3 by 3 by 3 by 3 by 3 by 3, and then we have 1 up here. And next one here, that be a total of 17 barnacles are there in this one. All right, again, the diagram is a little bit difficult for you to see. I could understand that. But again, when I'm finished, I'm going to zoom into it so you get a better viewing of it. But if you have this paper, you actually could see it and count it. Even on the original paper, it's kind of a little bit difficult to count. All right, so the number of barnacles is 6, 7, and 17. Okay, so let's go into the calculation here. Remember again, it's total over the number of samples multiplied by the area of the apparatus, which is 0.25. So here now, let's put this in the equation. So it will be the total number. So it'll be, let's just do the total right here first. Make it easier for us to calculate. So it'll be 6 plus, let me go back up here a little bit more. So it'll be 6 barnacles in the first sample plus 7 plus 17. And that all of that will equal to 30. Okay, so this will be 30. All right, so let's go into the equation now. So it'll be 30 over the number of samples, which is 3 multiplied by 0.25. So this will be 30 divided by 3 multiplied by 0.25. Okay. All right, and so all of this now will be equal to 30 over 0.75, because 3 multiplied by 0.25 is 0.75. Okay. So it's actually equal to 30 divided by 0.75. And so this now equals to 40. So it'll be 40 barnacles per, let me just put this slash right here. So for every square meter, so per meter squared. Let me put in the square by using, by writing that in. Maybe a little bit difficult for me to type that, and you see it as a square. Okay, so yep, so it will be 40 barnacles per every meter square. Okay, great. Now we have that. Let's go to the starfish. In terms of a starfish, let's jump into the top and see. Let's do some real counting here. For starfish, in this quadrat, there are 0 starfish in this one. In this one, there are 3. So 1, 2, 3. Okay, so notice 1 here, 1 here, and 1 here. So that's 3 of them in this one. Let's look at this one. We see a little piece here, right? But that's not even half, so we cannot even count this as it. So this will be 0 as well, right? So at least most of it will have to be in the quadrat for you to count it, all right? A little piece less than a quarter is really not significant to count. You cannot count that one, okay? All right, so let's now jump into the calculation here. Again, let's look at total quickly. So the total here is 4. All right, so a matter of fact, let's not even count that. That's 4, 4, definitely 4, okay? All right, no, sorry, 3 starfish in total. So there are 3 starfish in total. So it'll be 3, which is a total here, and all of them is only in the second quadrat, which is really not important. So 3 divided by 3 multiplied by 0.25, okay, 0.25. All right, and all of that now is equal to 3 divided by 0.75, because 3 multiplied by 0.25 is 0.75, okay? All right, and so all of this now is equivalent to 4. So here we have 4 barnacles per every meter square. Okay, let me type in the meters, write it in right now, real quick for you. All right, so it will be 4 barnacles for every meter squared. Okay, so that will be our answers there. All right, so let's jump on to the next part of the question now. It said, comment on what your answer in part B tells you about these 2 organisms in this ecosystem. So look at the number. The number of barnacles is 40, and the number of starfish is 4. So in terms of square meter. So what can you say about these numbers? What you notice with these numbers, 4 and 40? All right, so in terms of 4 and 40, you can clearly state that the number of barnacle is in the population is approximately 10 times greater than the starfish. Okay, let's write that in. The population of barnacles. The population of barnacles. All right, population of. Okay, let's go back a little bit. And get this back one little step further. All right, I think I have it now. Right. So the population of barnacles. All right. Population of barnacles is approximately 10 times. Okay, approximately 10 times, for 10 in 10 times greater than that of the starfish. Okay. All right. So about 10 times greater. So based on what you get by calculation, the barnacle population is about 10 times greater than that of the starfish. Okay. Again, it's a 1 mark question. All right. So let's go into this now. So name another sampling technique and state what it can be used to show. All right, so there are a number of sampling techniques that you can use right here, right? I'm going to see best I could give you most of them. All right, one of the easiest techniques to use is by observation, right? And I'm going to see, let me just put it beside it. And so observation will be one of the most common ones. All right, so let me just put this in red so maybe a little bit better for you to see. So observation, direct observation. So is it a standard ecosystem or the location or area and just observe? So you have observation. Okay. And observation is simply just to observe the organism in its habitat and you make a count. So observe and count. Okay. So observe and you count. All right. I'm going to see how best I could put all of them in or most of them. So you observe and count. All right. Observe and count. That's how you do it. All right. So again, the way it's used or what is used to show, you can state it right here. So how you do it is just to count the number of organisms, or you just observe, and you can observe for more than one thing. So you can observe behavior, you can observe the amount, you can just observe and count, observe and just make recordings or analyze the behavior pattern and all those type of stuff, the eating pattern. All right, so there are a number of things you can use to show by observation. Our next technique, again, I'm putting techniques here, and so again, just bear with how I'm doing it. So our next one is tagging. Tagging is becoming a popular one in that you will tag organisms and then release them, capture them, tag them, and release them. Sometimes you can actually use cameras with these taggings as well. And so the purpose of doing this, or what it show, is showing behavioral pattern. Okay, so you tag them to show behavioral pattern, or simply to observe their trends, behavior, and so on. Okay, so you can actually observe behavior patterns. Okay, all right. So behavior pattern. All right, and so let's go into a next one again. Let's look at another one which is called belt transect. Okay, so another one that, and this one actually used with the quadrats. Really, most times it's used with the quadrat. Not all time, but most times it can be used with it. So we have one called the belt transect. Okay. So the belt transect is used with the quadrat, and that will also show you number of organisms as well over a long distance. Okay, so it show you number of organisms. Let me show that real quick. Number of organisms over a long distance. So it's just like a piece of rope, and on that rope you could have specific markings in terms of intervals, and you can count the amount of organism between intervals. Okay, over a long distance. All right, so that's one there. We also have more, and so we have one that is called the net, and I think I've talked about this in a previous paper. All right, so let's call it traps. Okay, and so there are different types of traps. We have traps that are called pitfall traps, and you could go back to a paper that I did. You see the pitfall. You have pitfall traps. You have net. You can also use net. All right, so pitfall trap is one. You have net. Net is a trap. Also, you can use sweep net. So there's and when I say net, I mean you could use like those wire nets to capture fish and observe them and so on. Or I could use a sweep net. So it could be different types of net. Okay, so let's just make a note of that. The sweep net is a different one than just general nets. Okay, and the sweep net is what you use to capture organisms in grass and so on, or organisms that are really on foliage of plant, like the leaves and so on. All right. And this one again is used to capture and count number of organisms and so on, or the types of organisms that may be in a particular area, such as if you have corn, you may sweep the corn with the net and you capture the type of insects present, and you know the different species of insects or the number of them generally in a particular area. All right, and the last one I'm going to talk about here is fogging. This is not really a popular one, or it's not even a highly recommended one, a matter of fact, but fogging can be used. But what fogging does is that you use low dosage of pesticides, just to kind of get them immobile. And so when you have a piece of cloth, maybe, or some traps below the trees, and the organism that fall on those cloth or those catchment, you can actually count them and have an estimate of the number of organisms present in that area. Okay, so fogging can use to check the number of organisms present. All right, so great. So these are about what, 5? Yeah, 5 of them. Observation, which is direct observation, tagging, belt transect, traps, including pitfall traps, net, sweep net, and also fogging. Okay. All right. So that's that for this part of the question here. Matter of fact, the whole question is completed. So let's jump on to our next question. And for this question here, question number 2, I'm going to slight zoom into this. You can see the whole thing all together. All right. So that is great. All right, so this part here said define the term osmosis. Now, osmosis is a popular term that they ask you to explain or even have some application question to it. And so osmosis can be defined in a specific way. So there are some things you must include in your definition. So first I'm going to say it is the movement, and put that in blue. It is the movement of water molecules from an area of high water concentration. All right, a high water concentration. And when I say high water concentration, the word you can use for that is a dilute solution. Let me just put that in bracket for you. So it could be a dilute solution. Let me write it in short. All right, so you move from a high water concentration, which means a dilute solution or a high water potential, to an area of low water potential, okay, or low water concentration. All right, concentration. All right, so, and this would be a concentrated solution. We must put that right there, is a concentrated solution. So notice when I use the word concentration versus dilute, please, you know what I'm talking about. All right, so move it to an area of low water concentration, which means the solution itself is concentrated, and this must go through a semi-permeable membrane, okay? Membrane, all right? And so that's what osmosis is for 2 marks, okay? All right, so it must include the movement of water molecules, okay? And it must go across a semi-permeable membrane. It moves from high concentration to low concentration, okay? All right, so let's look at this diagram now. The question reads, it said diagram shows a plant cell in its original appearance before it is placed in a solution of sugar and water. It said the cell sap vacuole has a higher solute or sugar concentration than the external solution. So in other words, the solution on the outside is hypotonic, okay? Because it has more water on the outside. So on the outside right here is be hypotonic. Let me just write that in for you, hypotonic. Okay, so the solution is hypotonic. All right. Now, notice the particles, the water molecules are the smaller particles, and the bigger particles, those are the sugar molecules. And for this reason, again, they cannot pass through the semi-permeable membrane because they are too large, okay? because things go through semi-permeable membrane by nature of their size. All right, so let's go to the question, what the question asks us. And so the first part here said, draw an arrow on the diagram on page 4 to show the direction of water movement between the solution and the plant cell. So again, being that the solution here is diluted, solutions are dilute solution or hypotonic solution, it therefore means that water will tend to go what? Inward. So water move from the solution itself towards the inside of the cell. Okay, so that's exactly what will happen there. Okay. All right. So let's now jump on to the next part of the question here. I said, what term describe the condition of the cell after being in the solution for 24 hours? So if you place the cell in the solution after 24 hours, then the cell will become turgid because it gained water and the turgor pressure increases and so on. All right. So it becomes turgid. The word here is turgid. Okay. All right. All right. Now, the next part of the question state, it said the cell shown in the diagram in part B1 was then placed in a very concentrated solution, which is hypertonic solution now. So you move it from the hypotonic solution into a hypertonic solution. It said draw a diagram in the space below to show how it will look after 24 hours in this solution. So again, placed in a hypertonic solution, it will lose water. So in interest of time, I already drew this for you. So what will happen here is that water will leave the cell, okay? And because of water leaving the cell membrane will move away from the cell wall because of a decrease in turgor pressure. And the vacuole, as a result of that, also gets smaller, okay? So that's what will happen there for 3 marks, okay? All right, so let's now jump to the next part of the question. It said the table below shows some result from an experiment involving measuring the length of potato strips. Here now, you have the potato strips A and B. You have the initial length of potato strip, and that is per centimeter. That is measured in centimeter. So the original length of all the potato strips were 4 centimeters long. Then the length of potato strips after the experiment, so strip A increased to 4.4, and then strip B remains the same. So right here say that you have a percentage change in length of the potato strips. All right, so let's now go into what is saying. First part of the question says calculate the percentage change. So you're going to ask you to calculate percentage change in length of the potato strip. Okay, so let's go into that. All right, so we're going to calculate it. So what you want to do to calculate this is simple this. You're going to minus the final length from the original length from the final length. So the final length was 4.4. Okay, you're going to minus from that is 4. And the answer you're going to get there is going to be 0.4, okay? Now, what do you do with this 0.4 here? Let me put it on this side. Now, you're going to divide that by the original length. So let's put a little star for the calculation here. So it'll be 4 divided by, 0.4 divided by 0.4 divided by 4. And then you're going to multiply all of that by 100, okay? So, whatever answer you get right there, so let's say multiply by 100%. Let's put it right here. Multiply by 100%. Okay, and they have divided by 4. And so your answer here will give you 10%. Okay, so that be 10. So the percentage change will be 10. All right, so 10% change. And so right here now, put our 10%. So 10% change will be our answer. Okay. All right. So now let's move to the next part of the question. It says suggest what type of solution strip A and strip B were placed in. So by looking at strip A, there is an increase in length. And please, when you do an exam and they ask you to describe it, I know some person will say the potatoes grew. Please not say it grew. There's no growth taking place. Is an increase, an expansion. Okay, remember growth involves production of new cells. The cell get larger or the tissue get larger simply because they're taking water, not because of growth. Okay, so that's expansion. So because the potato in A increased in length. So we know that this solution here is going to be hypotonic solution, okay? So hypotonic solution. So it was placed in an hypotonic solution. All right, so just to make a point of that. All right, in cell B now, it remains the same. Nothing was changed. So this is the same concentration inside and outside. So it's called isotonic. So this is an isotonic solution here, okay? All right, great. All right, so let's move on now to the next part of the question. We finished with that. This is question number 3, and I'm trying to go as fast as possible, to save some time. So question number 3 is that the equation shows the process that occurs in living tissues, right? And results in release of energy. So energy is produced here. So once energy is produced, you know this is a respiration, right? But we're going to get into it a little bit. Now, the first part is the name of the process shown in the equation. Again, it is a type of respiration, but we want to be very specific with this, right? So this one here is called anaerobic respiration, okay? There's no oxygen used, so it is anaerobic, okay? So it's anaerobic respiration. All right, so what I want to point out to again, the specific type of anaerobic respiration in this case as well is also lactic acid fermentation. How we know this? Because when glucose is divided into two equal parts in terms of product, this easy way you could use to remember it, it is lactic acid, because lactic acid is C3H6O3, which is exactly half of the glucose molecule, okay? So this is called lactic acid fermentation as well, okay? Lactic acid fermentation. All right, just to make that point. Okay, great. So it is otherwise called lactic acid fermentation, or it is a type of anaerobic respiration. Now, it's a name the tissue in the human body in which this process most often occurs. And so lactic acid fermentation is generally in the muscles, okay? And it's a tissues. Now, just to point out this real quick. If the question asks where in the cell it takes place, it's a different question now. Where in the cell it takes place, then it will be in the cytoplasm, because remember now, anaerobic respiration takes place in the cytoplasm. But it asks you which tissue. So since it asks about tissue, then this must be what? The muscles, okay? In the muscles. All right, so great. Now let's move on to the next part of the question here. And I said, what is this process most like? It said when. Sorry, I said where. When is this process most likely to occur? When? And so on the two conditions you may want to put, right? So, but the basic idea here is that on the strenuous exercise. So during strenuous exercise, okay? So during strenuous exercise is when this normally occur, okay? All right. So during strenuous exercise, that's a normal time that this takes place, okay? All right, so not just on the strenuous exercise, but also when oxygen is low, okay? So if you're doing strenuous exercise and oxygen level is low, so when you have low oxygen, I just want to put that there so at least you can explain this if you need to explain. And so the body will go under a condition called oxygen debt, okay? And which means that the cells will continue to respire even if enough oxygen is not present. So it's during strenuous exercise, particularly when you have low oxygen. All right. Now, part 4 said describe how the toxic waste product from this process is removed from the blood. And so, what I want to point out here as well is that the waste product that they're referring to is lactic acid. Lactic acid will cause the muscles to cramp or to burn. Okay, and so a buildup of lactic acid, you get cramping in the muscles. And so how to remove it is actually think about how it is formed, and it's formed because of low oxygen. So to take it out of the body or get rid of it, you have to increase the level of oxygen. And that is why after an exercise, especially on the strenuous exercise, you breathe harder and also your heartbeat increase to pump more oxygen to the cells of the body or to the muscles of the body so you can get rid of lactic acid. You have to repay your debt of oxygen. All right, so here now, lactic acid is generally taken to the liver. Okay, so lactic acid is taken to the liver, okay, to the liver, where it is converted to either glucose or glycogen, okay? That's a general thing that normally happens. That's how we get rid of, come out of the blood, really. All right? All right, and so what assists this process, again, as I just mentioned, and this is aided, let me just make sure note of that this is aided by increased breathing and heartbeat, okay? So heartbeat will increase, and also so heartbeat will increase, and also your breathing will also increase. And simply because this is really to pump more oxygen again, more oxygen around the body, so the cells will have more oxygen, and the cells can now break down the lactic acid, okay? So without putting back oxygen, the lactic acid will not be able to break down, okay? All right, so that's that for that piece of information there. All right, and that's a 2-mark question. Let's go to part B. Part B now said the process shown by the equation at the beginning of this question and alcoholic fermentation in yeast both result in the release of energy. In fact, they're both respiration. Now, this one is asking us to write now a balanced chemical equation for the process of alcoholic fermentation. Now, so what I need to put here is glucose. All right, I'm not going to type this in. I'm going to write this in actually, because typing is going to cause me not to have my superscript, well, my subscripts really. So here I'm going to have C6, okay? So C6, H12, which is my glucose, okay? So H12 here. All right, I should have typed in this long time before. Anyway, just bear with me a little bit. All right, okay, so that will be our glucose. And our glucose will break down into ethanol. And so ethanol is C2, okay, H5, OH, okay? So that's my H5 there, and OH. All right, that's an O, okay? That's an O. All right, so OH. All right, and also they give you as well carbon dioxide, okay, which is CO2, okay? And then you'll plus the energy, which is ATP, or you can just say plus energy. I just put ATP, make it be real quick. So I'm put plus ATP. I can just say plus energy, okay? Plus ATP. All right, there we go. All right, so now, however, this equation is at balance, right? They ask for a balanced chemical equation. All right, you can go to one of my balancing lesson and you'll see exactly why balanced chemical reactions. Now I just want to move a little bit faster. And so what I'm going to put here is a 2 in front of this to make both sides of the equation equal. All right, and just to point this out, this is an uppercase C, not a lowercase c, it's an uppercase C, all right? This is because my difficulty in writing all of these things. All right, so that will be a balanced chemical equation right there, okay? All right, so now let's move on to the next question here. So name the energy containing compound produced in both of these processes and state 2 ways in which it is used in the organisms. All right, so the energy compound here is ATP. So it's ATP. Let me type that in real quickly. It is ATP, ATP. And ATP is the abbreviation for adenosine, okay? Adenosine triphosphate, okay? All right, so adenosine triphosphate is the word here, okay? Adenosine triphosphate. Now, how adenosine triphosphate is used is in many ways. And so typically, one of the main way you use it in the body or by organism is for movement, okay? So we use it for movement. In fact, generally speaking, for all the characteristics of life, any of those characteristics, that's what they use energy for, okay? So all the life characteristics or all the life processes, you use energy for those things. All right, and so movement is one. Also use them to transport substances around the body as well, to transport substances. And a matter of fact, I'm going to clarify this because it's not just any substances, okay? And I want it to be clear in terms of your explanation in examinations. The transport of substances, and this transport of substances is a process we call active transport. So when you're going through active transport, that is when it's required energy, okay? Another way you could use this is for chemical reaction, because guess what? Some chemical reaction requires, matter of fact, all chemical reaction requires energy. However, some absorb the energy, some release the energy. So typically for chemical reaction, so it aids in chemical reaction, okay? To aid chemical reactions, okay? So that's one there. And another possible one is for cellular processes, which I technically mentioned before, all the life processes. So cellular processes. And again, I'm just giving you extra information here, just in case you may see similar questions or you may want to explain or expound on certain question, you're able to do that. Okay. All right. Unfortunately, I cannot go through the whole lesson in terms of a topic, but you can always view the other lessons and go through them. And I know if you have questions, you could quickly ask them and I answer them as quickly as I possibly can as well. Now, question number 4, which is the last question on this section of this paper. So here now it said that species of all organisms show genetic variation among their individuals, right? And so here now it said lists 3 possible causes of genetic variation within a species, okay? Let me get a little bit of zoom into this so I can make this slightly bigger. All right, excellent, right there is perfect. Okay, great. So here we're going to name now or list 3 possible causes of genetic variation. And again, I'm going to list more than 3 for you, okay? Again, I really want you to know as much as possible can, or share as much as I possibly can with you. So mutation is a common one, okay? The mutation is a common one. Sexual reproduction. Yeah, because sexual reproduction, again, because of exchange in the genes or genetic material during crossing over, during meiosis, then we have variation within the offspring. Okay, so sexual reproduction. Yep. All right, so sexual reproduction would bring about variation as well among the species. Also, we can have the environmental effects or environmental changes. Okay, so we have environmental changes as well. All right, so environmental changes. We could have that as a next one, because changes in the environment could cause some organisms to look differently, such as if the temperature or the sun exposure, the type of food they eat, and so on. So let us put that as diet as well, yeah? So diet could also affect the variation among species. Don't put diet here. Diet is a good one because let's say looking at height or weight, based on our person, heat maybe will change their appearance, right? Their physical appearance, which is their phenotype. And so definitely diet could also affect the variation among organisms as well. Also, we have what they call gene flow. And let me just explain this real quick for you. The gene flow is really a phenomenon, right? Or a situation which new alleles are introduced in a specific species or region or area. So let's say, for example, you have moth, for example, right? A black moth and a white moth. They're living in 2 different places. And so for some reason, one of the moth migrate and then eventually find themselves in a different habitat or different region of that same ecosystem. And so they can now mate sexually, and then that mating could now cause a change in the appearance of the moth. So you can have a mixture of moth now. You can have white moth with black spots, or black moth with white spots, or even gray moth. So the gene flow moving from one region to the next region across the same species could actually change the appearance of some of the species or some of the organisms. Okay, so actually this is introducing of new allele or new genes within a particular species. And next one you could have here as well is well, yeah, let's leave it at that. So that's about 5 of them. Okay, great. So introduction of new species, new allele in that. So okay, let's stop it there. So those are the basic ones and general ones. All right. All right, let's move on to part B now. For part B, so the data below was collected by a horticulturist investigating height variation in a species of sod grass. So the sod grass now, if you notice here quickly, you have a category of height in millimeters, and so you have 120 to 130, you have 131 to 140, you have 141 to 150, and so on. All right. So we see the categories. The next column here is showing us now that we have the individual values for height in per millimeter. So in terms of the amount we counted, right? And so here we have the number of plants that were counted, or grass that was counted, and each of them with their specific values in terms of height. And then here we have a total number of plants measured in each category. All right, so for this one is 8. So this one will be 2. I could just quickly fill them in. Okay, so this one is going to be 2. All right, and this one will be 1. Let's just quickly fill these in. This one here is 5. Okay, yep. 5 is here. And the next one below here is 3, 6, 7. Okay, so we'll just fill them in. I know they're going to ask us to fill them, right? It's a name the type of variation being studied. So this is the first part of the question. Name the type of variation being studied in this investigation. What are we investigating again? We're investigating height. So if you're investigating height of grass, then height, because it can change over a large spectrum or a large range, then it is what we call continuous variation. Okay, so the type of variation is called continuous. Okay, so it's continuous variation. Okay, all right, so that's our 1 mark right there. So the next part is complete the table above by filling in the missing values, which we already done. All right, so we will we're good with that. All right, so let's go to the next part of the question here. And it said, plot a graph of your totals for each category on the graph paper provided. And I took the liberty in completing that before to save us some time. So I'm going to show you what I did and explain it real quickly for you. All right, so here now, let me go down to the graph. And here now you have the number of grass on the y-axis. All right, and we have the category here on the x-axis. And there's a point this out real quick. Let me put that in, that the height here is per millimeter. Okay, let me type in that real quick for you, per millimeter. Okay, so that's very important to put the units there. The categories again: 120 to 130, 131 to 140, 141 to 150, 151 to 60 to 160, 161 to 170. And we plot our graphs in terms of number of grass. All right, and we have that. And so if you notice the arrangement, okay, the 120 has probably the most amount of grass, so that was 8. Okay, all right, this thing I think drew up a little bit. I'm gonna pull it back down. It's supposed to be at 8. Let me see if it will work. No. Okay, I'll do it the next way. All right, I need to put it down here. All right, I know what I'm gonna do. All right, so bear with me. I want to put it down. It's a little bit too high. I'm gonna pull it back where it should be. All right. Excelente. All right, so that's where it should be. All right, so now we have this. No time for us now to go through and see exactly what they're asking us about this graph. Now, the question here said, how does this graph differ from the one normally expected for this type of variation? Again, this is a continuous variation, right? Which means among a species, you will see differences. Now, what is so different about this than other types of continuous variation graph is that the higher levels and the lower levels, right, or the higher or lower extremes, they are normally the lowest. But in this one, they are the highest. So let's go back to the graph and show you exactly what I'm talking about. So notice here the 120, which is the shortest type of grass, is a lot, okay, all the way up to 8. The grass that will be 151 to 160, they are the tallest grass, and again, those are the most. So the average grass or the less, which normally in a typical situation like that, the average height, for example, normally be the most, and the shortest and the tallest be the least, right? So the higher levels and lower levels normally be the lowest. Let's type that in. I'm going to demonstrate something for you real quick as well, right? So the answer for this is that the lower and the it said how this graph differ from normal graphs. So the lower and the higher extremes and the higher extremes, okay, and the higher extremes are normally the lowest, in terms of number of organisms, right? Normally lowest, okay? Normally lowest number of organisms, okay? So in this graph, the lower and the higher extremes are higher than normal, okay? So they are higher than normal. So you can I could put this here and say the low and high extremes in this graph are higher than normal, okay? So that's not normally supposed to be the case. In fact, let me just show you something real quick. Normally, under normal circumstances, your graph should shape like an inverted or upside down parabola. So once you're doing continuous variation, your graph should look something like this, okay? Which means in the middle of your graph, we have an average height should be the highest number, and then going left or right should be the lowest numbers, okay? So the most will be in the middle, and to the ends, higher and lower extreme should be the least. Because of course, you know, in any given population, the average is always the greatest number, and the extremes will be the one who extremely tall or extremely short, right? But of course, this graph is not showing you that. All right, if you notice, it comes down in the middle and then go back up to the end, which is not the typical way to see this type of variation. All right, so we finished with this part of the question here. As a matter of fact, we finished with this question, or finished with section A. So we're going to jump to section B. But before we go to section B, what I always want to remind you about is always, please, 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 read all 3 questions first, and then you start to answer the one that will earn you the most possible marks. Okay, look through the marks given and see which one will be best for you. All right, now we're here, and now we're going to look at now section B. And so again, you're going to answer 2 questions of 3 questions. Again, read through all the questions carefully before you start answering. In the interest of time, I've gone and typed this up, and so it's going to be easy to just run through. All right, so save us some time. So the first question, again, each question worth 20 marks, okay? And so let's do this in pieces. And so first part of the question, it said corn, which is maize, is a staple crop in the Bahamas. It's to explain what is meant by a staple crop. And a staple crop, that is a crop that is included in the main or regular diet, or the most commonly eat food in large quantities. So for example, your grits, your rice, and so on, those are staple crops. They are eaten very large amount in your main meal or your main diet. Now part 2 is a state 2 food products, okay, made from corn. And there are a number of products you get from corn: grits, cornmeal, cornbread, cornflakes, corn oil, corn syrup, okay? Even, well, I will say cornstarch, but cornstarch is not a food product, and so be careful of this. If we said products, then it could include cornstarch as well, right? But cornstarch is not a given here, because it's food products, and that's very important, things that you can consume. All right? But cornstarch, again, cornstarch can be used to make gravy. So again, it could be used in the kitchen. Okay, so I mean, guess we could use the cornstarch to make pastry, to make them more to create as a binding agents in your cooking. Okay, all right, so that could be a good one. But I trust if you put cornstarch, you can make a note beside it to say, okay, use binding agents in the kitchen, like making gravies, to make puddings and so on. All right. All right. So part 3. Here's identify 2 methods of preservation and 2 methods of packaging being used to extend the shelf life of these products. And so in terms of preservation, the access for 2, but again, I like to give you extra. So at least when you're going through the exam, or if you see these questions again, or similar questions, you know to approach them, or you have enough information to answer questions in different ways. Yeah. All right, so preservation, you could use drying method, take out the moisture so that reduce spoilage, add in chemical preservatives, That's a common way now, yes? Cooking or heating process. And cooking will kind of destroy microorganisms that will cause spoilage. Freezing, salting, canning, and also pickling, okay? So you may see the corn in the can, all right? And that is to preserve them for a long period of time. They may put even the little small baby corns in pickles, all right? And you may see them in the glass jars in the food store, okay? So that's another way to do it. And in terms of packaging, canning as well is a form of packaging. You may use vacuumed bags, which remove the air out of it. So a microorganism will not be able to survive in that condition without any form of air whatsoever. And you can also use airtight bags, okay? So those are typical ways that we can use to package these products. Now, part B, let's go to part B real quick. Now, part B is, it said, explain how farmers use chemicals to aid in the healthy growth and development of corn. And this is a 5-mark question. And it said chemicals. So of course you can list more than one chemicals. And so here I put them in 2 broad categories and kind of explain them. And so here I put, first one here is fertilizers. And fertilizers provide the necessary nutrients / minerals for growth and development. And I want to point this out that in most fertilizers, or generally in fertilizers, you have 3 main nutrients. You have nitrogen, phosphorus, and potassium, NPK, okay? Depending on the type of plant, the percentage NPK may varies, okay? Again, nitrogen is used for vegetative growth, which means the leaves and so on will grow healthy, green, looking good. Phosphorus is generally for strength and for growth in roots. The potassium is generally for flower and fruit development, okay? And pesticide is the next type of chemical. And again, how this is important in production of crops, because notice it here, to produce healthy growth and development of corn. How pesticide could help us to provide or to make us have healthy corn. And so pesticide reduce the destruction of crops by pests, including insects, fungi, and weeds, right? So you can use the pesticide to kill these pests that may destroy the crops. And so insecticides, I have them in 3 categories as well. Insecticides, fungicides, and herbicides, those are examples of pesticides. So insecticides may kill the insects that may eat and destroy the crops, because guess what? If, say, the corn leaves are being eaten by say, caterpillars, then the plant not able to photosynthesize properly to make its food that it needs to grow and develop. Okay, so getting rid of the insects will allow the plant to store its nutrients and use it for its own growth and development. Fungicide. Fungi sometimes will cause spoilage in fruits and so on, and even decay and damage to part of the plant as well. So you use the fungicide to kill the fungi that may possibly cause these destructive results. Herbicides is also critical. Now you can use herbicides, and herbicides are chemicals used to kill weeds or plants. And so you kill the plants that are not wanted. We call those weeds. So weeds are unwanted plants. And so you kill these weeds that are competing with the corn, and they compete with the corn for nutrients, water, space. Okay, because if you have weeds that are taking out of nutrients from the soil, then the corn may not get enough nutrients for its own growth and development. And of course, water is critical in this process as well, because plant will need photosynthesis and will need the water for photosynthesis to produce its food. Okay, and of course, space to develop properly, and also the space to make sure it have exposure to enough carbon dioxide and so on. So less competition is better for the plant itself. All right, so this part here, part C, it said describe 3 other techniques of modern technology, or modern farming, I should say, that have helped to increase corn production. And again, I have this in category, is a 3-mark question. And so the first one I have here is irrigation. Irrigation refers to water. And not just applying water, but automatic irrigation system. And automatic irrigation system could be used to ensure that crops have adequate water for growth and development. And I put at the end to maximize photosynthesis, because guess what? Without water, the plant can't photosynthesize properly to make its food. And it needs the food here for growth and development as well. And not only that, what is needed in high concentration or high amount to keep the cells healthy and turgid. Otherwise, the plants will wilt. And of course, without enough water to keep the plant cool, then the plant's stomata will close, no carbon dioxide can go in. And so there's a number of things we can talk about here about the importance of water. Okay, but that's a little bit of insight right there. Now, the next one is use of MGOs, which I already mentioned in one of the previous papers, right? And using MGOs, which are genetically modified organisms, all right? So genetically modified crops can be used to reduce destruction of pests or by pests. And also, you can use GMOs to develop more resistant strain crops against harsh environment. Because if you change the genetic makeup of these plants or crops or the corn, then they may survive better in terms of areas that you have more drought, areas that you have a lot of sunlight and less shading and so on. So it will provide the proper adaptation to survive in harsh environment. Okay, so you could change the genetical makeup of these crops also to store more water. So just in case you have a water shortage or a drought, the plant will be good to go. Also, you can develop these crops to produce larger fruits, right? And even faster growing crops as well, yes? So instead of waiting for months and months to reap your corn, you can reap the corn in shorter period of time. So you get greater yield, all right? Bigger fruits, greater yield. And so you save time and save money. So your production will go way up. All right. I'm not talking about if this is ethical or not at this point. I'm just saying how it benefits the production of the farm produced or the corn, what we're talking about now. The next one is machinery. Okay. And machinery, I'm specifically going to talk about modern machinery. And it's okay. Modern machinery can be used to prepare your land. So you can use for land preparation, such as the plowing and tilling of the soil. And so you save time by using persons or manual labor to do all of this, right? So you can use large tractors or special designed equipment or tractors to quickly plow the land, till the soil, and also to plant these corn. And also what is also important, their special machinery as well can be used to control the weed that are inside the fields. Of course, these again will reduce the competition for these crops. Also, what you can use it to do is the harvesting, to make harvesting much faster. All right. And so all of this together is pretty much reducing production time. All right. All right. So that's what we cover there. And again, machinery, use of GMOs, and of course, use of irrigation system. All right. And again, it's a 6-mark question. So part C of this question now state or reads, it's a name 2 natural threats faced by Bahamian farmers. And for 2 marks, natural threats could be a whole list of them. And so we have hurricanes as a possibility. Flooding could be a next possibility, which is generally unlikely, but it's possible. Okay, droughts, you may have droughts, long period of time without no rainfall. Pest, of course, will come and eat and destroy your crops. All right, and even livestock may even, if you're planting crops, for example, livestock could come in the field and actually destroy your crops as well. Diseases is another threat, and diseases could go for both plants and animals, right? All right. So now let's jump into the next question here. It's question number 6, and we're moving a little bit fast today. All right. So hopefully we could get to the end of it. All right. So this question refers to gas exchange in animals and plants. Now, part A asks us to define the term gas exchange. And gas exchange is the process by which different gases diffuse in opposite direction across an area or surface. So again, the term or the words used here, they diffuse in opposite direction. In other words, they're exchanging one for another. That's why the word gas exchange is in the statement. One gas is exchanging another by the diffusing across the membrane in different direction, okay? And this is possible because of difference in concentration. We get into that next time. All right, so part B now lists 4 adaptations of a gas exchange surface in animals which make it efficient for this process. And it's a 4-mark question. And the 4 things that will be there or needed is that the surface must be thin, must be moist, must have a large surface area, and covered with a lot of blood capillaries. All right? So those are the 4 things. And you can also think about surface like the alveoli or the villi. They are normally thin, they're moist, they're large surface area, and covered with a lot of capillaries. All right, part C of this question, it said the 2 diagrams below show 2 different structures used for the process of gas exchange, right? So diagram 1 will be A and B together, and diagram 2 is C, okay? All right, so now just jump in the question here. It says study diagram 1 and identify the process shown in picture A. State 2 reasons visible on the diagram to support your answer. So look at diagram 1 carefully. There's a little clue right there. If you notice, you'll see right here, you'll see 2 arrows going in, right? But I'm going to talk about that a little bit. All right, so let's jump down to the question now. The answer now is inhalation. You're inhaling, okay? So inhalation, and the evidence that are on the diagram, or the visible signs or reason on diagram. Look at the diagram carefully. Let's compare diagram A with diagram B. You'll notice that the area is larger in terms of the lung. So the lung volume is way larger. And actually, I could outline that for you real quick. And so I could do that. Let me just do that right now. Okay. All right. So, yeah. All right. So there we go. All right. And so here now you notice right here is a larger lung space. That's a lung space in the cavity right there, or the chest cavity. Is a larger lung space, definitely. All right, compared to the one in B. Very small lung space. I'm going to make outlines. You're going to see exactly what I'm talking about, right? The lung space is definitely smaller. So it's a larger lung space. And even look at the diaphragm. This is a darker line. The diaphragm is a darker line. So let's just go down real quick. Okay. So we have one larger lung area, a flatter diaphragm. We have a larger thoracic space or volume, okay, and the direction of the airflow. Now, the direction of the airflow, I'm not sure if they will give you a mark for that because they have the arrows there, but it is indicating that the air is going in, okay? That's why you know it's inhalation, all right? And the diagram is showing you that, so I just put it there so you can take your chances with that, all right? But of course, it is a good point because the arrows are showing you the direction of the flow of air or gases, right? All right, so part 2 of this now is that how does the process identify in C1 aid in the process of gas exchange? And this is the answer right now, is that because air high in oxygen, so the air that coming in is higher in oxygen and low in carbon dioxide, right? In terms of their concentration. So air high in oxygen and low in carbon dioxide concentrations rush into the alveoli. And it's due to a greater concentration of oxygen in the alveoli, oxygen will diffuse into the blood capillaries while carbon dioxide diffuse out of the blood capillaries and into the alveoli. Okay, so that will cover our marks right there. Now part D is a using diagram 2 only. Name the part C, and if you go back to it, which is a stomata. All right, matter of fact, stoma is singular, stomata is plural. So if you go back to the diagram, see right there, you notice it. Okay, so it is a stoma, stomata is plural, just to make a note of that. All right, and so the answer there is stomata. All right, and now part 2 now said explain fully how the size of structure C can be increased. So how can you have an increase in stomata? In other words, how can stomata open more? And this is the answer right here. And there's a 4-mark answer, right? So 1 is that water from the surrounding cells enter the guard cells. All right, the guard cells become turgid due to high water content in their vacuoles. And so as a result of this, you have a higher turgor pressure in the guard cells, causing the guard cells to curve. And this curving of the guard cell now will result in a larger or a wider open stoma. Okay, that will be your answer right there. All right, let me go back a little bit so you get all of it together. Okay, so please make a note of it. All right, go through it again. All right. All right, let's jump on now to the next part of the question right here. And this say explain fully how carbon dioxide is more likely to enter through structure C during the day. There's a 4-mark question again. Now, during the day, all right, so during the day, light stimulate the stomata to open. So the stomata open during the day, okay, because of the light. Now, light causes more solute to be in the stomata, all right. As a matter of fact, it's not in the stomata, in the guard cells. So I need to correct this real quick. So you have more solute will be in the stomata. Okay, so let's put that in real quick. Okay, hold a minute. All right, so here let me change this. So more solute will be in the guard cells. Okay. All right, all right, great. All right, will be in the guard cells. All right. So more solutes will be in the guard cells, causing more water to enter. All right. And so it said once the stomata are open, okay, once stomata are open, air containing carbon dioxide can diffuse through them easier or even inside of a leaf. Okay. So that's what you'll have there. Now, so during the night, now, there's a content with this to explain. So during the night, the water content in the guard cells, I have stomata here. So the water in the guard cells, okay, so the water in the guard cells decreased, and this result in closing of the stomata, causing less carbon dioxide to enter. Okay, so in the day they're open, in the day the stomata they're open, and in the night they are closed, and simply because of the water content. So in the night, the guard cells become flaccid, okay, and they will close. Now, last question here is that tropism and reflex actions are 2 ways of responding to stimulus, okay? It said distinguish between the terms tropism and reflex action for 4 marks. Again, I put a chart. Of course, you could do this in examination. No worries about it. It's easier for you to draw a chart and show the differences between 2 things. And the examiner will see it better as well, and you make your own checks to make sure that you have the correct stuff. It's easier for you to write and easier for you to see answers anyways, right? And so tropism here and reflex action. And so tropism is generally in plants, and reflex actions are generally in animals, right? Tropism is a slow response, and reflex action is way faster. Again, in tropic responses, it includes a chemical messenger, which is hormone. Okay, so the chemical messengers, they are hormones, but in reflex action, they are electrical messengers, and they are called impulses, right? Again, tropism, the response is through growth, so they respond by growing, but in reflex action, you generally respond by moving, and they move away from the stimulus. All right. Now let's go to the next part of the question, and we're almost there. Almost there. Now for this one, the diagram below shows a plant responding to a light stimulus. Okay, so the light is there, and the stimulus is there. Part 1 reads: state the name of the response shown by the shoot in the diagram. Now, what do you notice about the shoot? The shoot is going towards the light, correct? All right, and so since the shoot is going towards the light, then this is called phototropism, okay? Because tropism is a growth respond, and it's responding to light, so it is called phototropism. And please know the other tropisms, such as geotropism, okay? You need to know them, hydrotropism. All right, so you need to know all of those, right? All right, and so please go through and note them. All right, so part 2 of this question now state, it said name the hormone which causes this response and explain fully how this response is caused. 5 marks, a whole whopping 5 marks. So the hormone here is auxin. So auxin is the hormone. And how this response is caused, which means bending towards the light. If you go back to the diagram, it is bending towards the light. So I could go back up there real quick. The plant is bending towards the light, or bend towards light. So here now we can state that, hey, jumping too fast here. Yeah. So it's that when light is being directed onto one side of the plant, auxin will move to the darker side of the plant. In other words, it will be more concentrated on the darker side. And so auxin will be concentrated on that side. And because it's on that side, because auxin is responsible for growth, then you have more growth taking place. And a specific type of growth is elongate growth on the darker side. And this elongate growth on the darker side will cause the plant, so this will cause the plant now to bend towards the light. So the difference in growth, in the growth rate on opposite sides of the plant results in bending of the plant towards the light. Okay, and again, the darker side will grow faster and longer than the bright side. And so it will bend towards the light, okay? Now, parts of this question, so the diagram now below shows a reflex arc, right? Again, I'll label this for you real quick. So to save some time, we have the gray matter, which is A. We have B, which is a sensory neuron. We have C, which is the dendrites within the sense organ. Pretty much it's difficult to say exactly where it's pointing. So it could be the skin, or it could just say the dendrites in the sense organ. That could just be a sense organ, right? But I want to point why we label it as this, because I want you to know what is coming out of, or what is attached to the sense organ is the end of the neuron, which is the dendrites, because dendrites, they receive impulse, okay? So the dendrites, they receive impulse, okay? And so we have this location here is called the root of ganglion, and so the ganglion right here is where you find the cell body of the sensory neuron, right? Okay, so ganglion. And so here we have white matter, which is a part of the spinal cord. And so the inside of the spinal cord, the internal part is the gray matter, and the outer part is the white matter, just for you to know that. The opposite is true for the brain. The outermost part of the brain is the gray matter, and the inner part is the white matter. So you can notice, you could please make a note of that. Now we have the last neuron coming out of here is the motor neuron, okay? And at the end of the motor neuron, we have our nerve endings. And again, where the impulse will leave the neuron is always nerve endings. And so this is a muscle, and in the muscle you have what they call effector cell. And just to point that out, inside of the sense organ, we have what they call receptor cells, okay? So please make a note of that. All right, so it said name the structures labeled B and E and describe their functions in this response. And it's for 4 marks. So again, you're gonna name the structures, and that will be 2 marks, and then to describe their function is 2 marks, okay? And so B is our sensory neuron, and the function for the sensory neuron in the reflex action is to receive impulses from the sense organ. In this case, it is the skin, and transmit them to the spinal cord. Again, the sense organ could be different parts, could be high, all right? But the fact of the matter is, in this example, is the skin. So I put skin in the bracket for you, okay? And then now structure E is the motor neuron, and the motor neuron receives impulses from the spinal cord, all right, and transmit them to the effector cells in the muscle to bring about the action. And again, I refer to spinal cord because all 3 neurons are they meet in the spinal cord. So that's something for you to point out as well as to remember. Go back here. All 3 neurons meet in the spinal cord. The sensory neuron, the relay neuron, and the motor neuron, they meet in the spinal cord. And remember now, impulse move from one neuron to the other. Okay, so the relay neuron picks up information or impulse from the sensory neuron and transmit them towards the motor neuron. Okay, so that's to point it out as well. All right, so let's go down to the other part of the question now, and we're almost there. Now, yeah, we did answer that one. Okay, so part 2 of this now said identify where a synapse may be found in this diagram and describe what happens during during this response. And that's a 4-mark question. Now, just to make this clear, right? Just to make this clear, that a synapse is any location we have 2 nerves meet. Okay, so we have 2 nerves meeting, or 2 neurons, or 2 nerve cells. That's where you have a synapse. Okay, so you have it at the end of the of neurons. Okay, or between neurons. All right. And so here I have it as the end of B or E. And why I put B or E? Just for you to see. If you go back there, you'll notice that the ends of B and E in the spinal cord in the gray matter, you actually will see here a small space between the neurons. So notice where I'm over and over, you'll see that small space or small gap in the diagram. So let's just zoom into it a little bit. Maybe it can help you a little bit. Okay, so you can see now those small gaps between E and the relay neuron and also structure E. Okay, all right, let's dive down into our question. Let's go down in real quick. All right, let's go quickly into the next part of a question. All right, so we're almost there. All right, so here now the question has been answered. They said impulse flow to the end of the neuron, which triggers the release of chemicals called neurotransmitters. So neurotransmitters are released from the end of what they call the presynaptic neuron, which is the neuron it's coming from. Okay, and so coming from the presynaptic neuron, and in this case it would be, for example, the sensory neuron, right? And then it will now release or triggers the release of these neurotransmitters within what they call the synaptic cleft or the synapses. You can just say synapse, okay? And then what will happen next now? The neurotransmitters will cross the synapse and trigger the generation or production of a type of impulse in the dendrites of the other neuron. So in summary of it at the bottom right here, in summary, what we have here is electrical signals triggering a chemical signal, which are called neurotransmitters. And then the neurotransmitters then now trigger a next electrical impulse, which is the same as the one before. And so that will help to kind of boost and maintain how fast the neurons are traveling or the consistency of the impulse. So the brain now could interpret properly. Well, if it's a reflex action, then there's no brain involvement, okay? But just to maintain the flow or the speed at which these electrical signals are being transmitted. All right, so great. All right, so part D now it reads, state two ways in which a hormone response is different to that of a nervous response in humans. And I have a list of them here. You want to only access for two, but for hormonal response, it is the messengers travel in the bloodstream, while for nervous responses, the messengers travel through neurons. For hormonal responses, it's generally slower, and for nervous response, it is faster. The hormones, again, they are chemical in nature for hormonal. So the hormones are chemicals. The impulse, they are electrical in nature. Hormonal response are longer lasting, and nervous responses, they are short-lived. Okay. Hormonal response, they tend to be localized, okay, and nervous response tend to be general. A matter of fact, I have this on the opposite side, so I'm going to change this around for you because I'm going to change this around real quick. Because the nervous response, again, will tend to be specific. They can be very specific as well because, I mean, matter of fact, to be localized, both of them could be localized, a specific area, right? But hormones will tend to be general. So I'm going to change this real quick for you right here. Hormones tend to be general. Why? Because they spread all over the body inside the bloodstream, okay? All right, so let me just quickly change that. So it can be localized or widespread. I'm going to put right here. But generally, it is widespread because it travels all over through the bloodstream, right? And then nervous response tend to be localized, okay? All right. And so both of them, again, can be localized. And so the traveling of these messengers generally travel throughout the entire body, through the bloodstream, and then it will have an effect according to what is needed to happen. Okay, like example, growth hormone. All the cells in the body will produce new cells for it to grow and develop and so on, right? So generally have a widespread type of effect. All right, and for nervous system is generally localized because if you touch something that is hot, you move a finger, you feel the pain in the finger, or you just move a hand that is being affected by that stimulus. Okay, just to make clear. Also, here now that's the end of it, and here's where we say take care, and I hope to see you in the next review again. I will try to do some more before the examination. Again, thank you for being here. Keep on working hard. See you next time.